And I'm extremely happy to introduce David Krakauer, longtime friend and colleague from Santa Fe Institute. David started out in mathematics and computer science at the University of London, and somehow got derailed into the field of evolutionary biology by going to Oxford and working with Richard Dawkins and others there. He then came to the States and was a researcher at the Institute for Advanced Study in Princeton, working on theoretical biology. Then moved to Santa Fe Institute, where I met him, where he was a faculty member and then chaired the faculty. He got stolen away by the Wisconsin Institute for Discovery, which he's now the director of, which is part of the University of Wisconsin in Madison. He tells us it's quite similar to Portland in many ways. Then the Santa Fe Institute managed to steal him back. Now he's the incoming president of the Santa Fe Institute starting in July, which is really exciting for Santa Fe Institute. So before he starts, I just wanted to ask, he asked me, who is the audience going to be? And I said, I have no idea. And I invited a lot of people, as you can see. So how many people here are from system science? Okay. How many people from computer science? Mathematics? Biology? Neuroscience club? I know some people were supposed to come from that. Okay. Philosophy? Anything I missed? ECE?
Architecture.
Architecture?
Sociology.
Sociology.
Geology.
Okay, so you have your work cut out for you to talk about computation and evolved matter. All right, so let's welcome David.
Very good. Well, it's wonderful to be here. It's true. It's very. Yesterday, I was speaking at a conference of 500 librarians, which is very different and actually extremely interesting. The future of libraries, what they should be doing. So now, I'm very happy to make this more of a seminar. I thought there wouldn't be quite as many people, but I would be more than happy to be interrupted. I don't expect to finish. And so, in a way, given the nature of the seminar, I'm going to delve into some details, but I'd be happy to have philosophical conversations about what this represents as a research program, and I'm sure with the composition of this room, you'll have interesting insights that certainly we haven't thought about in terms of the future of computation outside of the domain of engineered devices, and whether this is even an acceptable metaphor, analogy, constructive framework. So I'm going to start with the quote that I sent in the abstract, which is so marvelous, from Alan Turing, when he said, we're not interested in the fact that the brain has the consistency of cold porridge. And it turns out Turing was very fond of those kinds of foodie metaphors. For the computer scientists in the room, remember old machines that had mercury display line memories, which is an acoustic memory system. Someone was nodding. The beautiful idea, actually my favorite memory, which essentially memory is encoded in propagating waves in mercury with piezoelectric crystals. Turing was the one who said he could replace the mercury with a more readily available English alternative, which was gin. So he was clearly a mischievous mind. In honor of Melanie, I'm going to give another quote from her PhD advisor, Doug Hofstadter, who says something very similar. He says, saying that studying the brain is limited to the study of physical entities would be like saying that literary criticism is focused on paper and bookbinding, ink and its chemistry, page sizes, and so on, and so forth. So this is a fairly prevalent style of reasoning about a certain kind of phenomena that we call computation, and the fact that it doesn't really depend on the materials. And of course, in these guys' cases, it's because they came from mathematics. Now, there's an alternative perspective, and one of the great expositors of the alternative perspective is A.A. Milne, who, because Owl said rather shortly, you and I have brains, the others have fluff. If there's anything to be done in this forest, and when I say thinking, I mean thinking, you and I have feathers don't cut it. And a classic perspective of this, from Epictetus, don't explain your philosophy, embody it. And there are names for this, of course, and philosophers know. The name for this perspective is functionalism, and the name for this perspective, and there are variants in extensive literatures on these perspectives called physicalism. But in fact, it does matter. And the question, I think, comes down to what extent does it matter? At what level of granularity does it matter? And that's what I want to go into. Just in the spirit of the sort of comedic, I couldn't help but note that there are versions of this by analogy in other fields. This is functionalism in film, where we're supposed to believe, I think, Russell Crowe looks like John Nash. This is what I call functionalism. It's just they have the same name. And over here, something a little bit closer to physicalism, that Cumberbatch actually does look a little bit like Alan Turing. And so clearly there's something going on here. When does it matter, when doesn't it? And in fact, Jess and I often argue about the fact that I believe that if you have an actor playing an English character, it should be an English actor. And Jess says, no, if Americans can do it, it doesn't matter. That's not the essence of what's required in acting, right? And details bug me. So I was a kid that didn't like to play with toys that were different scales. But just a personal story, when I met John Nash, and many years ago, not that many years ago, but a few years ago, and I had worked in game theory, and I introduced myself to him and I said, Professor Nash, honor to meet you. My name is David Krakauer. He said, Krakauer, hamburger, Frankfurter. And he's right, because they're all foodstuffs, by the way. If you go to Poland, Krakauer is a sausage. But then more importantly, the cities of provenance is a Jewish name, and the way it worked is that when you went to a city, rather than respect your name, they'd give you a new name, and it's your city of provenance. Krakow, Hamburg, Frankfurt. So he wasn't wrong, it was just a little weird. Okay. So I'm now going to be a proponent, this is the Santa Fe slide, for a middle way. Okay, so this is actually, for those of you who study Buddhism, will be familiar with the concept of the middle way. That's up there is Tibetan. And that's the Tibetan Buddhist word for the middle way. Of course, someone else can pronounce it better than me. You might have, I guess. And here's the original quote. It's interesting by analogy, again, what that meant, theologically speaking. Before the Buddhist period, there were two widely held beliefs. One was that all things were permanent. This described the transferring of the soul to a new body after death, and it's called eternalism. The other was nihilism, which believed in the extinction of the being after death. The Buddha did not preach either eternalism or nihilism. Instead, he taught the truth of the reality of conditioned existence. And that's what I'm going to talk about, conditioned existence. And it's quite interesting, by the way, that the convergence of theological thinking and thinking about consciousness, really, if you think about Kurzweil, and the post-singularity perspective, it really is this debate, much more than it is mechanism and function. And so there is an interesting convergence, even in philosophy, between these two perspectives. So this is all going to be about the middle way. And in that sense, my colleagues and I are phenomenologists. And that is, we believe that the way you actually come up with theories that are applicable in physical systems like biology or evolved systems is to study in detail, comparatively, a number of physical systems. And from that analysis, and from some exhaustive cataloguing of empirical reality, extract the regularities, and then construct your theory based on that data, as opposed to the opposite, which is in a sense what Turing did so successfully and arguably with greater consequence than any phenomenology. So I'm not going to claim it's more important, it's just our particular flavor. My last story, it's a Paul Dirac story for those of you who come from physics, is when Dirac was asked, how should one do science? And Dirac said, well, this is how one should do science. One should go out and one should conduct meticulous experiments and collect as much data as possible, put it in a database, analyze it statistically, compress out the irregularities, and present the world with a compact theory which maximizes the variability that can be accounted for by some model searching criteria. And he said, that's absolutely not how I do science. He said, I sit in an armchair and I say, if I were God, how would I design the universe? And he then discovered antimatter. So, you know, I don't want to be prescriptive. I think both are perfectly acceptable. It just happens to be that we are phenomenologists. And so, just a bit of background. It's interesting, if you think about computation like language in the biological sciences or in the social sciences, people approach it from different perspectives. So, computation outside of computer science, computation outside of computer science is used very often in neuroscience. That's the field, because of the influence of John von Neumann and his posthumously published book, The Computer of the Mind, that's most embraced the language of computation. So it's not uncommon at all to see in neuroscience textbooks people referring to the brain's computation. If you go to genetics, you don't see it at all. So in genomics, the word that's used is regulation. And that's because Jacob and Monod, who won their Nobel Prize for the discovery of the operon, were interested in cybernetics. It's a slightly different history. It's in itself a very interesting field. And so this came from Wiener and Ashby and Ross. And so that's an interesting challenge for us. Is regulation just another word for computation? And in social science, neither of those have really been used very much. It's much more within the framework of optimization, physical optimization. And in a way, I guess one of the questions that we have is, how do these fields relate to one another, and is there a synthesis that's empirically valuable? So, and again, I think taking colds to Newcastle, I should say Newcastle. Americans get it right, Southern English people get it wrong. You don't say Newcastle, I say Newcastle, because it's Newcastle. So the hardware-software distinction is one of those pervasive distinctions that you see in the literature. Obviously, we trace it back to people like Turing. Arguably, we trace it back earlier to formal logic. Maybe trace it back to Gödel and maybe others. But this idea that a computing machine is valuable, at least in the case of the universal Turing machine, of making a distinction between particular hardware implementation and the function that it maps, that it's computing, and the fact that you can feed it. If I'm assuming too much, just say, tell me, because I don't know, I'm assuming you know about Turing machines. If you don't, what should I do? What should I see? Nice enough. And what's interesting about this, by the way, is of course Turing, at this point in his life, wasn't really that interested in computing architectures. He was solving a problem in mathematics, the question of decidability, and that is, if you're given some statement made in a suitable formal system, can you decide whether it's true or false? This comes from Hilbert, and I think it's just a beautiful example for people like Governor Walker, who's the resident genius of the state of Wisconsin. Who helped move us back to Santa Fe. That, you know, ideas that could be about as remote from practicality as is imaginable, right? Formal logic have actually transformed the world in which we live, but on a time scale that is not available to So this is an interesting discussion, and one of the arguments that often comes up when we're talking with biologists about computations, brain is not a computer, the genome's not, what's hardware, what's software, that sort of thing. So that's the thing. And this is sort of how Turing actually did it, not in terms of dealing with that. The other way we think about computing, and this is probably how most of us want doing formal computer science think about it, is, well, you have some input and some data, and you have an algorithm that operates on the data, and in some appropriate time scaling, it terminates and generates an output that's hopefully correct. And that requires memory, in which provisional results are stored that can be drawn on in an efficient way. And that, of course, is really largely attributable not to Turing, but that sort of came a little later when people were trying to build, I think, and you correct me, a sort of a physical realization of a Turing machine. We now associate largely with jump-on learning methods. And that kind of perspective is actually used if you read neuroscience literature, for example. I mean, it's not uncommon. If you look at the study of perceptual pathways like auditory, olfactory, or visual pathways, that people will use that language. That there's an input, there's some kind of algorithm that's incarnated in spike trains, there is some kind of memory which is distributed and not obviously separable, which is interesting. And it generates an appropriate visual representation of the scene, a classification, for example. Someone walking the dog. And so on. And this is quite generally true in the study of perception. Because otherwise, the question I guess to ask is, if that's what you saw, beyond describing all of the nitty-gritty of the biology, how would you conceptualize it? And of course, through von Neumann and through David Marr, who made very explicit the kind of computational schema for interpreting perceptual phenomena. That's why I put that up. Because the question that we're confronted with, what is biology? What is it? What's the correct framework for understanding it? Okay. A completely different framework comes from cybernetics. And this is where regulation sits. And that typically deals with systems that have ongoing activity. So I put up a drone, because that's the current best example. And so imagine a drone, it's moving in a particular direction, and you perturb it. So there's some perturbation of the state of the system, and the system wants to regulate back to the course of destruction or the Amazon book delivery that it's charged. And there's sort of equivalent in our age. And the way that this is a rather famous paper by Conrad and Ashby, I think at the time of Illinois. And the idea was essentially that you have a system, it's engaged in some activity, there's some perturbation, that's D, and that changes the state of the system S, and that could be the rotations, it could be its orientation, its velocity, its altitude. And the regulator needs to be aware of the perturbation to the extent that it can then reorient the motion of the drone into the space of solutions which are accurate. So here the set Z corresponds to all possible states the system could be in, and G is a subset you think I that would successfully to Mary's house. And they derived a result, which is a very deep result, I think, called the good regulator theorem, and it basically says that the regulator, to be effective, and it comes down to essentially writing down the rank of the matrix, has to know essentially everything about the system in order to effectively ensure that the system behaves optimally. And it's stated like this, which is that the set of regulatory functions have to be with a set of correct solutions under a perturbation. And it relates, of course, to Kalman's ideas, which for those of you working in control theory, of observability, which is that you can infer the internal states of the systems by observing its outputs, and controllability, that is, the knowledge of the system can contribute to any arbitrary configuration that you desire. So where does the von Neumann-y thing, the Turing thing, map onto the Ashby-Conant-Wiener thing? And it's not obvious. The decomposition here is not obvious. I mean, you could draw little things, little sets around the states of the system and say that's the memory, and maybe these mappings of the algorithm. But it's a little forced. So I don't think, and I, again, defer to anyone in the room who does know this, there is an obvious correspondence at the moment in a kind of a formal language for talking about computing and the formal language of control theory. They're clearly related, but I'm not aware of a common language that incorporates both. Does anyone know what that is? No? Okay. Proof. Exactly, that's a kind of proof. It's a locally approximate realism. So the kinds of systems that we look at have the following properties. Mainly, they're very distributed, they're networks of various kinds, they're finite. Every system that we work on is finite and noisy. And that doesn't disqualify Turing-type reasoning, but it almost disqualifies it. And the notion of memory versus processing, almost impossible to disentangle. If you asked me to point to it, I couldn't really do it easily. I'll give you examples of this. And it's absolutely not obvious what the set of correct behaviors are versus the possible set of all possible behaviors. It's not something we know in advance. So we don't really know what is correct. And so notions of termination and so on become problematic. It's just, we don't know. And this is really tricky. And very system dependent. I couldn't give you a criterion. You say, I'm going to apply it, and it allows me to say in every case that I examine that that's software and that's hardware. So the language, if you like, that we've been given, that's immensely powerful in the world of engineered devices, is a little bit uncomfortable in the world that we work in. It's like a shoe that doesn't quite fit. It's still a shoe, you know, but it gives you blisters. And so, what do we do about it? And so now I'm going to give you phenomenology. Now I'm going to show you what we actually do. And so, I'm going to explain this a little bit more depth, because I noticed that there are only about 3 biologists in the room. And so I'm going to tell you a little bit about biology, but not too much. So, within the cell, there is a very ubiquitous enzyme. And an enzyme is a protein that modifies another protein, sometimes called a substrate, without itself being modified in the process. So that's sort of like the definition. And what it does is speed up rates of reaction. And one extraordinarily important class of enzymes are called the kinase enzymes. And they phosphorylate targets. They add a phosphate group covalently to an amino acid residue in a protein. And this thing turns out to be absolutely critical, because when it goes wrong, the number one pathology associated with the loss of disability is cancer. It is considered to be now the primary enzymatic mechanism behind long-term memory formation. It's sort of the absolutely most common and most functionally significant enzyme we know of. And what does it really do biochemically, for those who are interested? And I'll show you, it sort of switches the state of a functional protein.
Did you name the enzyme? Sorry.
Pardon me?
Did you say what the enzyme was?
Yeah, it's called a kinase.
Okay, thank you.
Kinase. And it essentially changes the electrochemical potential of the protein in order that it binds to something or does not. And this is just a drawing of a configuration diagram of, and I know it's sort of confusing, but it sort of says that there is a protein, it's called a MAP kinase, and it's a little bit of a confusing diagram. Forget this one for a moment. This is slightly better. You have something called W, it interacts with this enzyme, which is the kinase, and you spit out the enzyme unchanged, and you spit out the tagged target. That's what they do. They tag the target. And there's been an awful lot of mathematical biology working on the chemical kinetics of these enzyme systems. And one of the things that was realized quite quickly is that by just changing some rate constants, if you have a population of kinases, you can produce at the population level something that looks like a switch. So it looks a bit like a transistor in the aggregate. So what you have here is those sigmoids are, if you like, the quantity of W star against the normalized concentration of E. Okay? And so it's sort of intuitive, right? That if I add more of this kinase enzyme, you produce more tagged target. But by just changing the duration of the association of the complex when the enzyme hasn't been transformed as target, or when it's living with it, you can move between what looks like a fairly smooth input-output function to what looks almost like a digital switch. And this was extremely interesting to theorists because they thought we've discovered the enzyme that is the transistor in the cell. That was the idea. They're ubiquitous, they're everywhere, and in fact that is in some sense what they are, because they're used to filter input-output functions everywhere you find them. Hence, it's not surprising that they're implicated in so many different functions.
What's the time scale?
So
And
this is
how fast will it
microsecond.
switch?
Microseconds, milliseconds.
Is it a reversible?
It's completely reversible, and there is another enzyme called the phosphatase that rips the tag off. And in fact, it's the tension between the kinase moving forward and the phosphatase moving backwards that gives you these kinds of input-output functions. It was later realized that if you add a feedback to the system, it becomes more like a toggle. So now, the trick is that you turn, I've changed from W to K, but now I have a phosphorylated target, and that now becomes a kinase itself. So it autophosphorylates. Right? And when you do that, you can keep this thing on permanently. So now it's not just an input from the environment that you're modifying, but the system itself can modify itself.
So it's feedback.
It's feedback, it's positive feedback. And hence you can build memories. This is the key here. So this, with a little bit of energy thrown in the form of ATP, becomes a very long-lived memory. Okay, and the reason why this is important, by the way, is that this thing can K3 and so on. These are the little switches you find all over the synaptic density, which is the thing that we associate with correlated with long-term potentiation. So if you knock out these switches, you lose your memory. And so that's a very reductive statement I made, but it's just that somehow this component is very strongly implicated in how we do it. So the problem with all of these models is that they are mean field. They're ODE. So we decided to study them in their full stochastic glory. And I won't dwell on this, but just to say that I think this is how you have to go about thinking about computational biology. If people assert, this one's already left, which is good, success, eventually there'll be enough seats for everybody. That's my objective function. With some despair. And so you write down these equations which essentially tell you what the hopping probabilities are between different modification states of a target. So I can phosphorylate once, twice, 3 times, 4 times, 5 times, 6 times, and I can take those modifications off. So this is actually a kind of a multi-switch. It exists in a variety of different states, from 0 to n. And when you write those kinds of systems down in detail, you discover all sorts of glorious behavior. A variety of different phase transitions, critical behavior, the kinds of world that we like to get into, which is the kind of statistical mechanics of a simple protein that's ubiquitous in the cell. And it exhibits interesting tunneling phenomena and so on and so forth. But the point is here, for you to bear in mind, is we play with systems that exist at realistic concentrations, and you get a huge amount of noise and a huge amount of uncertainty in terms of the state. So you have probability distributions that give you the sense of where the system lives, but very little certainty. So that's just one system, and there are departments that do nothing but this, study these kinds of enzymes. Okay. You get all sorts of interesting physics. So that's one unit I just wanted to tell you about. Now, where do the concepts which I started with, hardware, software, correct, incorrect responses fit? Well, you could say, well, maybe the protein itself is the hardware, and the network of interacting enzymes is a circuit, and that network is not hardwired. It depends on the state of the environment. So, for example, if I'm not tagged, I don't connect to Jess, but if I am tagged, I do. So I can start building up a graph, an interaction graph, which is something like a circuit in the cell, which depends on the particular state of the environment. Okay? Who I connect to when is hardwired, but it's context dependent. Okay? So maybe this is the sense in which we could refer to programmability. Maybe that's what we mean. But it's sort of limited. It's not a universal programmability. But it's not a calculator. There is an I/O function, which I've told you about, which is they act as filters of some kind. amplifiers as well. And their true functional output is realized in fleshy things, like inducing a cell to divide or not, or causing a cell to die or not. So, I guess the question I have for you, a philosophical question, is, do you really give a shit about any of this? Right? Because I'm using this language of computation and I.O. Does it really make any... Is this just a kind of deodorant that we apply to the phenomenon after the fact? And what really mattered was writing down the dynamical systems, the stochastic dynamical systems, which actually respect the microscopic reality, which provide us with intuitions about how we should intervene in a real physical system. So that's sort of my big question. Yes, Mary?
So in computer science, we usually start with the functionality. That's what we're interested in. And figure out how to get to that functionality via these kinds of tools, right? Or building blocks.
Yes.
In biology, I presume that it's basically the same thing. You want to understand, well, you want to understand what is the functionality. That's the thing you want to understand, right? So what you, the equations that you just didn't give us that, right? Or do they?
Well, do they? I think they do. I think in the end, the equations which represent the conditioned existence, to use the Buddhist phrase, that respect the particularities of the molecules that we're studying, are the things that we need to understand to be good biological engineers. And the larger language of computation at this level doesn't seem to give me very much to use. I mean, it's not a practical tool. And hence, by the way, this distinction that happened very early on in AI, and it's happening again now in computational biology. I don't mean biologists that use computers and this kind. Aren't we really in the business of writing down stochastic dynamical systems? And the computational loss is not helping. And especially given the fact that theorems, even Les Valiant and others who claim that they're operating in the biological space, it's so far removed from the particularity of the systems we work on, it looks like a forced metaphor. I'm not saying I believe that, I think that is the discussion. And so when people say cells are computing, in what sense? Do they mean Turing? No. Do they mean Ashby-Conant regulation? No. In what sense? And that's what I sort of want to understand. And we're going to get there. I'm going to build up examples of mathematical models that maybe in aggregate point at the new kind of computation perspective. But that's the key. And you can play this game and say, well, maybe these are where those variables map. It doesn't matter. Let me give you another example. And I'm going to bore you because I actually think that this is where science lives. It's at the level of the detail and you can't ignore it. And the onus is on us to abstract away the specificities in our theories, but to explain the natural world, if that's what your stated profession is. So here's another system, and it's something we work on, it's epigenetic. So again, I'm just going to explain what this is for those of you who don't know. There's a lot of mystery around this word epigenetic. So it basically means the following. You all know that we have a genome, and the genome is made up of nucleic acids. And that breeds true, and you transmit that modulo a few mutations to your offspring in recombination as if you're human with your sexual partner. Okay? Now, those nucleotides can be modified. Okay? And those modifications also can be propagated to your offspring, but not as stably. So they don't typically last multiple generations. An example of that is a methyl group, methylation. Now, more interesting than that is that DNA is not naked in the cell, it's supercoiled, right, with proteins, and these proteins here are called histone proteins, and the DNA is wrapped around them. And these proteins that you sort of coil DNA around have tails. They're called histone tails. And those tails can be modified. They can be phosphorylated by a kinase enzyme. And there are numerous different enzymatic modifications of the tail. And a few years ago, we calculated that the information bearing capability of nucleosomes is about an order of 10 greater than the underlying DNA. Okay, so this gets at that. So what are the functions of the tails was the question. So this is how it works. There are enzymes that write to the tails, and they change, for example, electrostatics, or they change binding affinities. And so that was, in the early days, it was thought that the only role played by the nucleosome was packing. I mean, in fact, up until about just over a decade ago, 2 decades, it was just packing. But we now know that there are also erasers that can remove the modification, and there are readers. It's actually much closer to a Turing tape, by the way, than the underlying DNA. Much closer. And in fact, it's even linear in the way that it operates.
Semi-infinite.
Pardon me?
Semi-infinite.
And semi-infinite. Yes. And so the way it works is you have this enzyme, it recognizes modification on a tail, it reads it, and it can propagate it. It can go left and right. And it says, okay, the neighboring nucleosome should have the same modification, and so on. And that's how it propagates up and down the DNA molecule. And what is it doing? We can talk about that at length. At my institute now, we have a very large group that does nothing but that. It's a very big field because, once again, it's implicated in everything. Obesity, cancer, you name it. So why am I telling you this? So just skip that. There's a problem with the epigenome. In 1865, a very famous meteorologist called Mendel, he was a meteorologist. He studied with Doppler, who was a physicist. He taught physics at a high school. And not that it matters, but this is a kind of covert remark about transdisciplinarity. He wasn't a geneticist, they didn't exist. Okay? And worked out what are called Mendel's laws of independent segregation. And he tried to explain how, over the course of time, certain characteristics could be really true. And I sort of have these in the wrong order because I need to show you something. This is what me and you, but I'll show you it already because we're smart enough to be able to remember one slide back. And so imagine that we have a DNA molecule, and it has all those nucleosomes in it, and those are tails that have been modified. So just think of it now as binary. So when the daughter cells divide, the way it works is those little spools, they get independently segregated to daughters. So one daughter cell will have it, and the other daughter cell won't. Okay, is that clear? Because it's one chain, there aren't duplicates of the nucleosomes. And the problem is that you want the daughters to remember what the parent cell knew. But in fact, if you just propagate this forward enough time, you exponentially will lose signal. Right? Because every division you'll lose information from exactly one parent. And so on. And this was actually in the 1950s a great mystery of genetics, how you could propagate forward indefinitely without losing information under an entropic force known as mutation. And this was Schrodinger's great book of his life, was him wrestling with this anomaly, which is how can a complex structure be propagated indefinitely without it ultimately terminating. And so it was proposed, a number of different mechanisms were proposed, and all of these are associated with various Nobel Prizes. And the one that won was the so-called semi-conservative mechanism. Okay? And the semi-conservative mechanism is that each of the strands contains enough information to replicate itself. This is just template-based copying. There are a variety of ingenious error correction processes that ensure that the integration is not correct. But with DNA, you've got it because you've got a duplex, right? Because you have two copies, you can more or less ensure that the information is propagated reliably. But with the epigenome, you can't do it. And so the great mystery of the current time with the epigenome is how on earth does it get propagated? It's not known, and yet it's now known that almost every environmentally salient factor that you encounter is encoded in the epigenome, not the genome itself. Okay? Because you can't change the genome in your lifespan, right? But you can change what gets turned on and off, and that comes from the epigenome.
So are you saying that Lamarck was right?
Well, it's interesting actually. So there's a few things to say about that. The first one is most of this is developmental time. So when cells divide within a single generation, the modifications are preserved across cell generations. There is a special kind of epigenetic change which is conserved across generations, and that's methylation, which I mentioned. So notice these aren't changes that I drew to the DNA itself, but to a protein that's associated with the DNA. And that doesn't go through the germline. So the mark required germline transmission. But methylation is much closer to the marker. But there's more required for the mark to work, it turns out. But nevertheless, it's very close. So this is like developmentally the marker. And so how could memory work in such a system? And so this is what we're proposing, and it's really ripping off another paper that John Hopfield wrote many years ago to try and account for memory in neural-like systems. And I'm sure many of you know this paper, and a classic paper in neural networks. And the problem that John was working with is this one. You say, okay, I have some letters that I have to identify, but they're corrupted, and I want to tell you what the right letter is. Right? And the metaphor, and actually the physical model he used, which we were talking about this morning, which is the discrete maximum entropy model, and the Ising model, is generating an energy landscape where all of the letters are stored at the bottom of the wells, and so this will configure into the right configuration for these solutions preferentially when starting somewhere on a slippery slope. And so a perturbation to a level that's not massive will tend to be attracted back down into the attractor of this dynamical system, this Hamiltonian, whose output then is the letter that you design. And this is called content addressable memory. Computer scientists know about this. Be very different from RAM type memory, right? So you're trying to retrieve an address typically in content addressable memory, not find something at an address. So we thought, well, maybe that's more like the memory of epigenetics. It's not like semi-conservative replication. It's closer to a neural memory. And the basic concept is as follows, right? Now, the adjacency matrix that we write down in the Ising model, that comes from dendrites and axons with neurons, comes from this propagating wave of the reader-writer enzymes on the linear chain of the DNA molecule. And so that's what connectivity between non-adjacent sites means, is that a mark on a tail here can be propagated to a tail there by a sort of a cascading enzyme traveling down the DNA molecule. And you can write down a series of graphs and you can do calculations. And again, I got too much on this side point. But just again, diagrammatically, what does this mean is if these were the right epigenetic states that you wanted to propagate, and each daughter cell was binomially sampling from the nucleosome to the parent, how would I reconstitute the full complement set? Well, you do it as long as the perturbations aren't too great by using, in a sense, redundancy, which is encoded in the adjacency matrix. That's the basic premise. And you can show, doing all this stuff, and again, it's too much over this, that you can actually get almost 90% recall with nearest neighbor connections. So you don't have to assume unrealistic from the point of stereochemistry of molecules, what's connected to what. Almost perfect numbers. But of course they're less than one, and so you probably end up with a bit of it. But cell division is finite. And that's actually quite important. And I think that's why, by the way, the inheritance system of the organism is genetic, and the inheritance system of the cell is epigenetic. Yes, it works. So again,
I just didn't hear what you said. You said that the inheritance system of the cell and the inheritance system of the organism are, and then I missed the word.
Oh, I'm sorry. Yeah, because the interesting point about these systems is that they are still noisier than the semi-conservative templated mechanism of DNA replication.
Right.
And so it does put an upper bound on the number of generations where you can reliably, within some limits, propagate information. And because cell generations are strictly limited, so cell divisions in your body, I don't know what it is, it's something like 15 to 20. Within that range of divisions, you still have quite reliable information propagation. But if evolution had used epigenetic mechanisms, we wouldn't be here. And so, now again, what's hardware? Well, I don't know, enzymes. Again, we have a kind of circuit of a sort because we can write down our adjacency matrix, which reflects the read-write rules, and so on, right? And again, you should be thinking this way. If I came to you as a computer scientist and said, look, here's a mechanism, it's absolutely critically important to complex life, it's absolutely critically important to disease, here's a mathematical model that captures some elements of it, what does this look like as a computational system? And, well, it looks a bit like quantum addressable memory, is what it looks like. But this is what I want you to think about. Because this is, in a sense, the future of this kind of work. So, last one, Jess is here, and Jess did much of this work together, but any hard questions you orient to Jessica. But I was going to show you an example now on a totally different scale, that's social. And you'll hear again, people say, societies compute solutions, right? So, what is the price of that good? Well, we're a distributed population. Each of us has an estimate of the value of the good, and through a mechanism like an auction, we come to some equilibrium in terms of our estimated value. And as you all know, auction systems are designed so as to produce a price profile that suits either the buyer or the seller. The same is true with voting. You can say, well, we want to come to a reasonable outcome as to who has transient power within our societies, and society computes, given a set of preferences, a voting pattern that ideally results in the right candidate being elected to office. With no editorial remarks about Wisconsin. And so, which is of course an editorial remark, guys. So this is where the kind of data that we have. So Jess is the expert of this world, but essentially you have a bunch of a time series with individuals engaging in conflict. It doesn't matter what species, we have very highly resolved data for non-human primates, we have very highly resolved data for humans. But the essential idea is that over the course of time, you have a number of labeled individuals engaging in conflict. So this is me, Jessica, and Melanie. And the next time it's Max and Andrew. Okay. And what we're interested in, given that observed data set, is both encoding it in a very parsimonious form, and given that encoding, reproducing statistical properties of that time series out of sample. And so here's an example of placing that behavior in a Markovian lattice, where you say, okay, I could have a rule which says, if Melanie has fought in a previous conflict, then I'm going to be agitated and fighting the next one with the same probability. So this is the class of rules which is individual-centric, but we label that one way. These are the higher order correlations that cannot be factorized. Okay, so everything down here in the matrix says they don't reduce ultimately to the product of the 1-1 rules. So it implies some kind of what we call synergy, but it doesn't matter. And so, for example, here's a rule that says 2 individuals had to have fought in the previous round in order that 3 individuals would fight in the next. And you can see that puts demands on your memory, because you have to remember who was fighting. And it places some kind of demand on coordination, because you need to know who to fight with in the next round. Okay? So this is a space, a natural, basis set in which to represent the Markovian strategies that we're going to induce from the data set. Which is what you do, and again, But you do this actually in a way that perhaps wouldn't be familiar to people working with machine learning, which is that we know that individuals are severely cognitively constrained. So there are classes of model that are inadmissible given our prior knowledge about their cognition. Now that's a dangerous game to play, because humans